Well, good afternoon, everyone. Welcome to our the climax of our neuroprosthesis seminar series this year. And we're extremely lucky to have a distinguished speaker, Vivian Mushahwar from the University of Alberta, an old friend of the FES Center and the Neural Engineering Center, has been in this field, well, as long as I have, let's put it that way. So a long time. So Vivian has a Bachelor of Science in Electrical Engineering from Brigham Young University, a PhD in Bioengineering from the University of Utah, did postdoctoral fellowships at Emory University and at the University of Alberta. Currently, she is the Canada Research Chair in Functional Restoration. She's a professor in the Department of Medicine and the Division of Physical Medicine and Rehabilitation. And she is an institute director recently, right, in the Sensory Motor Adaptive Rehabilitation Technology, or SMART, and is a network director also for Smart Technology Innovations. And she has about this many other appointments, which I'm going to skip right now. But thank you so much for coming today, Vivian, and let's welcome our speaker.
Thank you very much, Bob, for this very kind introduction and for the invitation to be part of the neuroprosthesis seminar series. It's really a great pleasure to be here, and it's just been an absolutely fabulous day meeting with so many people. So thank you, everyone, for taking the time to spend a bit of your day today with me. So probably some of you would know that the overall goal of the work in my lab is to develop interventions that would improve function after neural injury or disease with a specific focus on spinal cord injury. And those interventions are primarily neuroprosthetic associated interventions in which we use electrical stimulation in innovative ways to enhance function and to prevent secondary complications. So what I'm going to focus on today is work in which we tap into the spinal cord in different ways to improve the ability to walk after spinal cord injury. And towards the end of the talk, I'm going to touch on a new project that in which we are looking at improving arm function by improving the bimanual coordination between the arms. All the work that I am presenting today, I should do this, I guess, is done at the University of Alberta, and I'd like to acknowledge that the University of Alberta is on Treaty 6 land, which is the traditional lands of First Nations and Métis people. I also would like to acknowledge that, where should I be? Where is the control? Where should I be aiming? There. Okay, so I'd also like to acknowledge the amazing group of researchers that I've had the great pleasure of working with over the past many, many years. And as Bob mentioned, we used to be the SMART Network, the Sensory Motor Adaptive Rehabilitation Technology Network, but we are now the most recent institute at the University of Alberta, and we are the Institute for Smart Augmentative and Restorative Technologies and Health Innovations, and the abbreviation is iSmart. Our members come from many different disciplines, including computer science, many aspects of engineering, health economics, many aspects of medicine, neuroscience, nursing, nutrition, rehabilitation, social science, textiles, and even consumer needs. And we've come together for the purpose of developing intelligent medical devices and innovative rehabilitation interventions that would prevent injury, restore function, and augment the capabilities of persons with diverse abilities, with the intention, of course, of improving quality of life. And we're doing this through interdisciplinary collaborations that engage the full spectrum of research, healthcare, and industry, modeling very much after what CASE has been doing for so many years with the neuroprosthesis program. Now, iSmart is organized in 4 cores that are highly integrated, and they're really focused on accelerating discovery and its translation into clinical and social impact. Some of the activities that we do actually is a wide range of activities that span from cell cultures to numerous preclinical studies focused on rehabilitation innovations and neuroplasticity, to robotics. Not quite sure if that, yeah, there we go. Some of those are working. So artificial limbs and making those artificial limbs very intelligent in terms of the user-device interactions, innovations in rehabilitation, wide range of robotics, from surgical robotics to exoskeletons to for the legs and for the arms. We look at augmented reality and innovations in telerehabilitation for service in remote environments. The iSmart also has a business arm. It is also a University of Alberta entity, and this business arm is ST Innovations, or Smart Technology Innovations, that is led by a very capable business team, and we are guided by an industry advisory board that is made of seasoned national and international entrepreneurs and advisors. And what our goal is with SC Innovations is to empower health innovators. So, and basically help them to remove some of the barriers in the pathway to commercialization and implementation of innovations. So we do that by offering health innovation companies, entrepreneurs, and innovators multiple services in preclinical engineering, and also connections to end users and clinical expertise. We also use a multidisciplinary approach to all the projects that come in from industry. And with that, we help early innovations to be better positioned for commercialization and entering the marketplace. And with our, sorry, I always want to keep pushing these arrows, but I really should be pushing this. So that's okay. I will learn. So what we do with, by helping innovators to advance their technologies through the development and commercialization strategy or journey, we also upskill highly qualified personnel from industry. So we bring them in and we train them on specialized equipment, specialized processes. And the idea there is to diversify the economy in Alberta and allow for upward mobility. Now, for work in my lab, as I said, I focus on spinal cord injury. And of course, as you all would know, depending on the level of the injury and depending on the severity of the injury, we have different ramifications and functional consequences of spinal cord injury. So you've got the incomplete paraplegia, and of course that's, the complete paraplegia where the legs are paralyzed. You've got incomplete paraplegia, you have complete tetraplegia, and you have incomplete tetraplegia. What I'm going to be focusing on today in my work is on the restoration of ability to walk. That is primarily for people with paraplegia. And then at the end of the talk, or towards the end of the talk, I'll be talking about improving arm function, and that's primarily targeting people with incomplete tetraplegia. So the type of activities that we do in my lab, especially for walking and improving arm function, very much focus on activation of spinal networks in innovative ways to restore these functions. And this is what I'm going to focus on today. But I'm just going to show you a couple of other things that we do in the lab. And one of those is looking at improving assistive robotics, developing and making assistive robotics more intelligent. This includes exoskeletons, and I guess a sideways upper limb exoskeleton to help with arm movements. And we also look at smart walkers that would improve function for persons with impaired mobility. We also look at developing wearable bioelectronic devices to prevent secondary complications. One example of that is Smarty Pants or Smart Electronic Pants, and this system prevents the formation of pressure injuries. This received FDA approval a year and a half ago under the name Prolivia, and in fact, this is being trialed at the Cleveland Clinic as we speak right now. We also have developed another wearable, just as an example, and this is a sock or the smart ongoing circulatory compressions. And the idea here is to prevent deep vein thrombosis, edema, and improve circulation in general. So this is just a general overview of the kind of work that we do in my lab. I'm going now to focus your attention on the activation of spinal networks for improving walking and arm function. I'm going to start with walking. And before we do any projects, we try to understand what are the principles that we need to meet and say that this is a successful accomplishment of that task. And for walking, we know that we needed to meet the guiding principles, the clinical guiding principles for restoring functional walking. And basically what this was is that the movements that are generated need to be predictable and coordinated. They need to be weight-bearing so that the weight of the body is carried by the legs, not the arms, during the stance phase of walking. The walking needs to be propulsive to allow for forward progression, and it needs to be fatigue resistant to allow for reasonably long distance of walking. So these are the principles that we have set aside to meet for restoring walking capacity. And I'm going to start off with persons with complete paraplegia. I'm going to call this a severe spinal cord injury that leads to either motor complete or full complete paralysis of the legs. And for these conditions, we have over the years focused on activating this region of the spinal cord, or the lumbar region of the spinal cord, which is known to be the spinal cord center for locomotion. As all of you would know, this is a very small region, the spinal cord. It's the lumbar enlargement. It's only about 5 centimeters right at the base of the back. But the reason we're interested in it is because this region contains a large proportion of the neural networks involved in locomotion. So we've been interested into tapping into this region for the longest time. And before we tap into it, we tried to understand where are those locomotor networks, how are they organized within this lumbar enlargement. So to make a very long story short, we've done over the past 20 years a lot of mapping studies trying to understand where those networks are localized. And what we found is that they're primarily localized deep within the spinal cord, deep within the ventral horn. Of course, you're all familiar with spinal cords, but what you're looking at is a typical cross-section of the spinal cord with the butterfly being the gray matter. This is the back, this is the front, and you've got the different meninges. So the question here was, how do you activate these locomotor-related networks? Of course, what you would know is that one way that has been demonstrated even for severe spinal cord injury is the use of epidural spinal cord stimulation. And again, in this crowd, I don't have to describe this surgical procedure, but the idea is that you're putting an array of electrodes on the back, the dorsal surface of the spinal cord, which is the lumbar enlargement. And as you would know, what this would look like, so if we look at the array being represented by green over here, being placed on top of the dura mater, when we stimulate through this array, what you end up with is activating dorsal roots, and the electrical stimulation kind of penetrates about 200 to 300 microns into the dorsal surface of the spinal cord, and then this indirectly activates the locomotor networks. And as you would know from the beautiful records that have been out, is that with epidural stimulation in persons with severe spinal cord injury, either motor and sensory complete or primarily motor complete, across different studies, different countries, University of Louisville, Mayo Clinic, and in Lausanne by EPFL, people with severe spinal cord injury receiving epidural spinal cord stimulation coupled with extensive physical therapy have been able, for the first time, to with epidural stimulation, to stand and take steps over ground. And the amount or the duration of walking was anywhere from, or the distance of walking was anywhere from 90 meters to 230 meters, depending on the type of stimulation strategy you use and the type of rehabilitation intervention that you couple that with. This, we thought, was exceptionally impressive. These individuals were unable to stand before or step over ground, but with epidural stimulation, they've been able to achieve this task to a certain extent. But our question was, does this walking match the clinical guiding principles for functional walking? So, and it's debatable whether they do or not, but we thought perhaps we are able to do a little bit better if we were to activate those locomotor networks more directly. And that is by placing penetrating electrodes into the spinal cord with tips that to directly target these regions. Of course, this is a more invasive, hi Hunter, this is a more invasive, this is a more invasive approach. So the question is, will it be able to provide improvements in functional walking? So, and we needed to understand what kind of walking it could produce. So just to give you an idea, the concept of intraspinal microstimulation is that you have this microarray of penetrating electrodes that is placed within the lumbar enlargement, and we pattern the stimulation through those electrodes so that you are able to activate motor synergies. And we primarily look at synergies that are necessary for walking, such as an upward synergy, a forward synergy, a downward synergy, and a backward synergy. So the key synergies are involved in walking. So that's the general concept. And of course, this implant is blown up over here. It's a very small implant. So to ask this question, if we can really do that, we developed a micro implant that looks like this, that we place in the lumbar enlargement. This micro implant is composed of hair-like microwires that penetrate the spinal cord with their tips reaching deep into those intermediate and ventral regions of the ventral spinal cord. We have then the leads traveling on top of the cord, come up, and they are anchored to the spinous process nearest to the lumbar enlargement, and then they're connected to a wireless stimulator that generates the patterns of stimulation that we're interested in. And we've been doing this for quite some time. This is the longest going project in my lab. And what we found out is that the system, at least in small and medium-sized animals, this is more rats and cats, appears to be stable, safe, and could produce functional movements. So what we found is that in animals implanted with the system for up to 6 months, at least 80% of those microwires continue to produce the same movement again and again and again, suggesting that the implant is stable. And by the way, this was implanted in animals that did not have a spinal cord injury, allowing them to move around and do whatever animals do, just to see how stable the implant would be. We also found out that in these chronic implants, that the microwires themselves and the intraspinal microstimulation that we're putting through them causes minimal tissue damage. Obviously you're putting something in the spinal cord, so there's going to be damage associated with the insertion of the wires, but it seems like it's limited to that, suggesting that it's safe. And then we found that looking at standing, we found that with intraspinal microstimulation, you can get practically 5 times the duration of standing that you can get with peripheral stimulation. And in our hands, in the same animals, it was with intramuscular stimulation, and had appropriate and stable posture, suggesting that it could be functional, at least for standing. But of course, our interest has been in overground walking. So can we produce overground walking with intraspinal microstimulation that meets those clinical guiding principles for functional walking? So what we did, these experiments were done in cats, and we did is we implanted those microimplants in the lumbar enlargement of the cats, and then developed control strategy for walking that very much mimics the natural control strategy for walking. And of course, these strategies are strategies that are used in Cleveland and done very well with the implants. But basically what we've done is we had a feed-forward mechanism, so feed-forward oscillations, as we call it, where the stimulation basically cycled through the different phases or the different synergies of walking. Remember, we're looking at flexion, forward, downward, and backward synergies, and that basically simulated a central pattern generator, if you want to think of it that way. And then we had feedback-driven modifications of the durations of the phases for these phases and the magnitude of activation within each phase. And this was done based on information from the location or the position of the limb in space, as well as the amount of force that was generated within LM. As I said, these experiments were done in cats. So this is what the experiment looked like. These were done in deeply anesthetized cats. The implants were placed in the lumbar enlargement, and then the cats were placed in a sling that you may or may not be able to see clearly over here. The sling supported the trunk and the head of the animal, and the forelimbs were put on a panel that was just a little bit elevated over this walkway. The legs were free to move. This walkway had force transducers all throughout that allowed us to also measure the amount of force that is being generated with each step. And what you may not be able to see, there were small sensors on the legs that told us where the limb is in space. Okay. Now, this is what whoops, that's not that. Let me see, can I get this to, no. Okay, let's do this. Okay, so this is what the walking looked like. This is now, the goal of this experiment was to see how many times the animals can cross this 3.5 meter walkway. And one thing that I should mention is that this walkway has unpredictable friction. That meant that it could be easy or it could be difficult to walk, depending on how those wheels of the cart wobbled against the pathway. So we had no idea what the resistance is going to be, and the controller had to adjust to this resistance because it basically simulated going uphill or going downhill. Can the controller deal with these changes so that the walking remains stable? And the idea again was to see how many times the animal can cross this walkway. And what is incredibly exciting, other than the fact that the walking looks near normal, we of course both legs are walking, but we can only film the one side. But what was incredibly unpredicted at that time, we were hoping that the animal would walk, I don't know, 100 meters, 200 meters, is it was able to cross this, the animals were able to cross this walkway 300 times. That is equivalent to a kilometer of walking. So this was incredibly impressive. And we looked at what this walking looked like from the perspective of force generation. And what you see for the equivalent of 1,000 meters, we have the forces that were generated during those steps were basically stable throughout the duration of walking. In our hands, when we did the same comparison with intramuscular stimulation the walking basically decayed about 150 to 175 meters. What this told us is that intraspinal microstimulation is doing something about the recruitment of motor units in a way that is a little bit more natural, and possibly that is the reason why fatigue resistance is seen. And I'm more than happy to talk to you about the evidence that we found about this, about the near normal recruitment of motor units. Now, with these results, we found that those were very exciting, but also very compelling, that perhaps there is something here that we could translate to humans. But we can't go from small animals to humans right away. We needed to go through an animal model that more closely represents humans, and that is the pig. So the pig, among all larger animals, tends to be the best model for translation, at least for this kind of work. And that's because, not only because the spine and spinal cord of a pig has the same dimensions as that of the human and the same morphology, but in the pig, just like a human, unlike sheep, unlike monkeys, there is a large subdural space. Just like in humans, there's a bit of about 5 millimeters of space between the dura and the surface of the spinal cord. So that meant that this was really the appropriate model to use to translate to humans. And over the last few years, I would say over the pandemic, we started really investing quite a bit of time in modifying the intraspinal microstimulation implant so that it would be suitable for a large animal and hopefully suitable for humans in the future. So this is now a zoomed in image on what the implant looks like and the different components of it. We have the microelectrodes with the sharpened tips. Each microelectrode has a lead that has a microcoil, and this microcoil was put there to adjust for the large strain, the large changes in the length of the spinal cord as the animal moves, and similar to what happens in humans. So we needed to account for that strain. Then these leads all bundled together for another fixation here through a PDMS tap or a silicon tap, and then they are coiled together to produce a coiled cable. This cable exits the dura and is attached to the dura, and then it is attached to the spinous process to stabilize that, and then attached to a stimulator. So this is really what the full system looks like. You've got the implant over here. Of course, is sitting on a slide over here, glass slide, which is released at the time of the implant. Then we use basically Medtronic extension cables to connect the coiled cable to the wirelessly controlled and implantable stimulator. So we've done quite a bit of acute testing in pigs with this stimulator, with this system, and modified it over the last, I would say, 3.5 years. And what we're now doing is implanting it in pigs with intact spinal cords. And the idea is to see how this system would function primarily in terms of its safety and in terms of the recovery of the animal from the surgery. And this is now an example. You hear a little bit of noise, but this is an example of one pig the morning after the surgery. You could see that this is, again, neurologically intact, so it's able to stand up and walk around and seem to be functionally stable. But what we definitely, so we were actually surprised by this fast recovery from surgery. We had not anticipated that. But we need to be tracking this over time and see what's going to happen to the implant and the spinal cord over time in these large animals. So this is still early days, but that's where we are at this point. Now, I'm going to switch gears a little bit, and I'm going to focus on our approach to improving walking capacity for persons with incomplete paraplegia, and it could include also people with incomplete tetraplegia in that category. But for this work, what we had proposed is that what we need to activate is not just the lumbar enlargement, but we really need to activate a large portion of the spinal cord by engaging the arms simultaneously with the legs in a rehabilitation intervention. Okay, and our thinking is that this engagement of networks across the spinal cord is what's necessary for functional walking, especially after incomplete spinal cord injury. So why, do we think so? Well, we all know that we swing our arms while walking, and this arm swing is not just the arms are not just mechanical appendages that are passively swinging during walking. Arm swing is neurally driven, and the drive to the arms is coordinated with the neural drive to the legs. Now, this information is not new. It's been around for a long time. And there have been quite a few studies that have shown that activity in the arm modulates the activity of the legs. So our question is, why isn't that knowledge built into current rehabilitation interventions? So interventions that are currently available for walking tend to be more gait-specific and focus primarily on the legs and not a whole lot on the arms. So our question now became, will we be able to improve walking more than what's currently possible if we engage the arms simultaneously with the legs in a rehabilitation intervention? And because we wanted to make the intervention something that is easily translatable and something that actually could be implemented even early after spinal cord injury, we decided to have people sit and cycle. So what we decided to do is simultaneous arm and leg cycling with the leg movements assisted with electrical stimulation. So the cycling would provide coordinated activation of the arms and the legs, and the electrical stimulation not only would assist with movement generation at the legs, but would also send a sensory volley back to the spinal cord that is physically appropriate and timed well with the generation of the movement. So the question was, this was radical, because if you want to train walking, you don't train cycling. That's what rehabilitation tells you. So the question became, will this actually improve walking capacity, and how much would that improve walking capacity relative to existing interventions? So we recruited study participants who had an incomplete spinal cord injury, AGSC and D. The level of injury was anywhere from C4 to T10, chronic injury, and we had two groups. We had one group that only cycled with their legs, assisted with electrical stimulation, and the other group cycled with their arms and legs with the same electrical stimulation delivered to the legs as with the group that did only leg cycling. And training was for 12 weeks for this group. So I'm just going to show you videos of what this looks like. Here is an example from one person who was part of the legs-only cycling group. And what you could see is their walking pre-training and walking post-training. And what you probably will be able to see is that not only is the person able to walk much faster than after training than before training, but they're having less difficulty in swinging their legs. So the walking is taking less energy to achieve. Now, here's an example of a person in the arm and leg cycling group. And here again, you see significant improvements. Before cycling, you see that the person was hunched over, basically walking on his toes because of spasticity in the legs, and he was looking at his legs all the time while walking. After training, this person is standing upright, not looking at his legs anymore, having much less effort in swinging his legs, and certainly walking a whole lot faster. He's not looking at his legs because he's had sensory perception now, so he regained some sensory function in his feet, so he knew where his legs are. So the take-home message is that cycling does improve walking. So there is a transfer of function from cycling to walking. The question is, how much is this improvement? How am I doing in time? Okay, good. So how much is this improvement? To do that, what we did is we looked at a whole bunch of studies that had gait-specific training and had kind of a training intensity and duration that was similar to our study. And what you see over here is a collection of these studies, and what you see on the y-axis is the change in walking speed. And the numbers over here are the numbers of study participants in each of these studies, and the line over here shows the average across all of these studies in terms of the absolute improvement in walking speed. So now, if we look at the results from our study, remember our study was pilot, and now we're trying to extend it to a much larger study. So our numbers are smaller, but what's intriguing is that the leg cycling group has improvements in walking speed that are similar to the walking speed gained from gait-specific training. So perhaps this could be, an easier approach to train walking. But if you look at the group that did arm and leg cycling, the improvements in walking speed were at least twice what we saw with the legs-only cycling group. So, so I guess the message here is that, again, including the arms along with the legs may provide with better improvements in walking speed, and we see the same thing with walking distance. So, again, perhaps what we have over here is a more effective therapy that requires less expensive equipment, less dependence on physiotherapists to perform the intervention. Now, in addition to looking at those clinical metrics, we looked at the biomechanics of walking, and what we saw that in both the arm and leg cycling group and the leg cycling group, we saw improvements in preferred walking speed and in stride length. But what we also saw is that only in the arm and leg cycling group, we have a whole bunch of additional improvements in walking biomechanics. So the question is this only a mechanical thing, or do we have something happening at the nervous system level? So we first looked at the excitability of the corticospinal tract after training to understand how that activation from the brain to the spinal cord is changing as a function of the intervention. And of course, as all of you would know, one way to look at the corticospinal excitability is to use transcranial magnetic stimulation. When you stimulate the region of the cortex, you're able to produce a motor evoked potential. And what we were looking at is the peak-to-peak amplitude of the motor evoked potential before training and after training. And what we found out is that there was an improvement in motor evoked potential, in other words, enhanced excitability of the corticospinal tract in both groups, but only the arm and leg cycling group reached significance, and that was double the improvements in peak-to-peak amplitude. Another thing that we looked at is what's happening to the message going to the spinal cord, and to the muscles. How is muscle activity regulated before? How was it regulated before, and how is it regulated after the training? And what we found out, and here this is we just looked at one muscle. I'm using one muscle as an example over here. I'm looking at the EMG activity of the tibialis anterior muscle during walking. And this group, doesn't require me to go in great depth over here, but you see that there are periods when the activity is high and periods when the activity is low. This is now from neurologically intact study participants. Now, here's an example from a study participant who was in the arm and leg cycling group before training. And what you see is, this is now mean and standard deviation. You see that their walking pattern is the regulation of the activity to tibialis anterior is very much different from what you see during the normal regulation, and you have activity in places where it shouldn't be. Now, after the arm and leg training, This is the same study participant. Now look at their average in the blue. And what you see is that now the pattern of activation is starting to follow the normal pattern of neurologically intact study participants, suggesting that there has been a regulation of muscle activity, specifically after arm and leg cycling. So that is super exciting, telling us that there are neural changes that are taking place, and it's not just biomechanical changes. So now what we have started relatively recently is seeing if we can further enhance the improvements in walking capacity after incomplete spinal cord injury if we use transcutaneous spinal cord stimulation. And this is very preliminary, and we're going to show you just one slide in terms of what we have over here. What you see is a person with incomplete spinal cord injury walking before training, and then now it's only been 6 weeks of training. And probably what you would notice is that the person is not hunched over the way they are over here, and they're certainly walking at a much higher speed. So the question is, would 6 weeks be adequate? If we add transcutaneous spinal cord stimulation to arm and leg cycling, it's unclear. This is just very early, but we're seeing that perhaps we are enhancing the activity within the spinal cord, perhaps enhancing that propiospinal connectivity between the cervical and lumbar regions. And with that, augmented with the arm and leg cycling, perhaps we are engaging more of the spinal cord and improving walking capacity. So just to summarize this part of the talk, I hope that you would agree, and I think this group would agree, that activation of locomotor networks in the lumbar enlargement may restore walking capacity after severe spinal cord injury, and walking capacity that perhaps would meet the clinical functional guidelines for, or clinical guidelines for functional walking. I hope you would agree that engaging networks throughout the spinal cord by coupling the arms along with the legs in an activity-based therapy could improve walking after incomplete spinal cord injury. And we think that transcutaneous spinal cord stimulation may be an effective means for modulating networks further throughout the cord. Transcutaneous spinal cord stimulation still has a long way to go for us to understand how exactly it works, and we're doing a lot of that in a parallel pathway in our lab. So for the last few minutes, I would like to just talk about a very new project in the lab in which we are interested in improving arm function after incomplete tetraplegia, and basically focusing on improving the coordination between the two arms. So Cleveland has led the way in restoring arm function after severe spinal cord injury. We are looking at incomplete spinal cord injury. And here from the many study participants who come to the lab, of course, they identify arm function, those who have tetraplegia, as a very important component that they would like restored. But one of the things that they told us is that they would like to do better coordination between the two arms. So we thought maybe we could look into this component, and we wanted to understand how transcutaneous spinal cord stimulation applied to the cervical level could play a role in enhancing bimanual coordination. Okay, so we did these experiments in neurologically intact study participants. We always start studies with neurologically intact study participants. And what we had them do is sit in front of the kin-arm exoskeleton, and the kin-arm exoskeleton allows for bimanual movements in the planar plane, in the horizontal plane. We asked them to do 3 different visually guided elbow extension movements with and without the application of transcutaneous spinal cord stimulation to the cervical region. Each movement was repeated 20 times, and then we recorded movement trajectories and EEG activity, but I'm only going to focus on the movement trajectories for now. Okay? So for transcutaneous spinal cord stimulation, this is where it was applied. We gave or delivered 40 hertz stimulation trains. Pulses were 1 millisecond long, and within each pulse there was a carrier frequency of 10 kilohertz. So the movements that we asked the study participants to do are here. We asked them to do unimanual movements, so just they needed to reach with their dominant arm from a home position to a target. We asked them to do bimanual dual goal movements, where the two arms needed to reach to a target, to two targets simultaneously, to two different targets. And then we asked them to do bimanual common goal movements, where the two arms had to collaborate to move from the home position to the target, and so that this ball sitting on a bar does not fall off. So if they didn't, if the two arms did not collaborate, the ball would fall off. Okay, and they, just as a reminder, the movements were performed with and without the application of cervical transcutaneous spinal cord stimulation. So I'm just going, let me show you first just the raw trajectories that we looked at. And I'm going to tell you how we looked or assessed the biomechanics of movement. We basically looked at two things. We looked at, so these are just examples, these are examples from one person for the different movements. We looked at movement error. Basically, we looked at the deviation of the movement trajectory relative to a straight line from the home position to the target position. And we looked at movement time, which is the time that it took to move from the home position to the target. Okay, so that's how we quantified the biomechanics. And I'm going to tell you what the result was. Interestingly, with transcutaneous spinal cord stimulation, there was improvement in the biomechanics based on these two metrics, but only in the common goal movements. So this is what it looked like. This is now the different movements, the bimanual common goal, bimanual dual goal, unimanual movements. You have no transcutaneous spinal cord stimulation in the solid bars, and you have the transcutaneous spinal cord stimulation with the hash bars. Very interestingly, the error was significantly reduced when transcutaneous spinal cord stimulation was applied, but only during the bimanual common goal movements. These are the movements where coordination between the arms was necessary. The movement time also had a strong trend, didn't quite reach significance, but a strong trend in terms of reduction in the time that it took to move. And again, only in the common goal movements. So something is happening with transcutaneous spinal cord stimulation. The question is, and remember, these are neurologically intact people. So the question is, what could be contributing to these improvements, and only in these kind of movements, the common goal movements? Question was, or one possibility is that transcutaneous spinal cord stimulation is doing something about proprioception. So we wanted to understand the effect of transcutaneous spinal cord stimulation on the sense of position. So the way we did this is, here again, we use the Okay, let me see. All right, let's do this. Here again, we use the kin arm exoskeleton, and we had the robot. Now the people are not seeing their arms, so this is completely without visual feedback. And the robot would move the arm to a certain spot, and the other arm has to be, the study participant had to match that position or the mirror of that position. And you would move to the robot will randomly move to 9 different positions and repeat them multiple times, and the person needed to match these positions. Okay, so this is what the results looked like, or at least this is a raw data from one study participant. This is without transcutaneous spinal cord stimulation. The green is what the robot did, and the cyan is what the person did. Of course, you know, this is a mirror image of the movements. And with transcutaneous spinal cord stimulation, the same person did this. So this is what it looked like. And what we did is we quantified the endpoint distance error. So the difference between this point and that point is what we really quantified. This is super preliminary, and we have an application, a grant application on this that we hope will get funded. But if you look at the group, what we found out is that, and this is mean and standard deviation from 10 people, that with transcutaneous spinal cord stimulation, there seems to be a significant reduction in the error of position matching. So the question is that what's happening? When you're coordinating the two arms, you have to have an enhanced sense of position so that the two arms move together. Is that what's happening? Perhaps that with transcutaneous spinal cord stimulation, you have this enhanced sense of position. Still preliminary, but we've done this in 2 study participants with spinal cord injury and now incomplete cervical spinal cord injury, and it seems that perhaps fits. Too early to know. But I'd like to end on that and conclude by saying that, repeating some of the summaries from earlier, that activation of locomotor networks in the lumbar enlargement may restore functional walking after severe spinal cord injury. Engaging networks throughout the spinal cord may improve walking more so than what's currently available. Get specific training after incomplete spinal cord injury. Transcutaneous spinal cord stimulation may be an effective means for modulating networks throughout the cord, and perhaps cervical transcutaneous spinal cord stimulation may improve bimanual coordination of the arms. So what are we doing now along those projects? As you saw, we're testing intraspinal microstimulation in pigs. We are assessing the effect of transcutaneous spinal cord stimulation combined with arm and leg cycling in persons with incomplete spinal cord injury, as I showed you, and this work is continuing. And we're continuing to investigate the effect of transcutaneous spinal cord stimulation on proprioception. And our goal is to, really assess the effect of transcutaneous spinal cord stimulation on bimanual coordination on people with incomplete tetraplegia. And our hope with that project is that perhaps we could develop a rehabilitation intervention that would further improve arm function for persons with incomplete tetraplegia, combined with activity-based therapy and transcutaneous spinal cord stimulation. With that, I'd like to acknowledge the amazing group of people in the lab, so, trainees and support staff who did all the work that I presented today, and the great group of collaborators from the University of Alberta and beyond who were involved in the projects that I presented today. And of course, I would like to acknowledge the general support from many funding agencies that have made it possible for us to do the work that I presented. So thank you so much.
Well, thank you, Vivian. We have a little gift as a remembrance of your trip here to Cleveland and the FES Center.
Thank you so much.
We can have that shipped if you want.
It's okay.
So, questions for our speaker? Yeah. Oh. Am I going? No, Chris, start.
So I'll quickly ask. So, thanks for the nice presentation. For the cycling study, I'm curious if you have tried yet or plan to look at whether the addition of arm to cycling increases the intensity, or do you try to have similar intensity, maybe measured by heart rate, across the 2 interventions? So is it really critical that the arms are involved, or is it just increasing the intensity of exercise, and that's really what drives the effect that you see?
Yeah, that's a brilliant question. And at this point, we've evaluated cardiovascular function, but not in enough people to really tease that out. But what we do know is, from a neurological perspective, you really are seeing those differences. So is that because more of the body is engaged? Perhaps. And more of the spinal networks are engaged? We'd like to say yes. So in the studies that we're doing right now, we are being much more specific in measuring heart rate and a perception of exertion. So we stop every 10 minutes or so and get those measures to make sure that we are quantifying this better. But I don't think that it's only a metabolic thing or a biomechanical exercise effect. I think there is, it's possible, but there are definitely neurological improvements. Yeah.
Thank you for the talk. I was intrigued with the video you showed with the arm and leg training that the person not looking down to their feet, and you said that there was some enhanced sensation. Can you elaborate on that? Was it the result of including the arm and leg, or is it just that the result of that a stimulation that happened because of the leg movement?
Yeah, excellent question. I took out some of these results just because of time, but actually the improvement in sensation, we thought at first was significantly better in the arm and leg cycling group than the leg cycling group. What we found out is some of our participants in the arm and leg cycling group had lower sensory scores. So when we took at the beginning, so when we took that as a co-variable, what the initial sensory scores were, there was no significant difference between the legs only cycling and the arm and leg cycling. So we believe that improvement in sensory function you would also see in the legs only cycling group because both groups improved in their sensory perception. This particular person specifically looked down on his legs all the time. And I was quite amazed when I walked in for the final assessment because he was standing upright. So I thought the harness was pushing him up or pulling him up. And that wasn't the case. He was actually able to stand up and not look down because he had improved sensation and he had reduced spasticity in his triceps surae, in his gastroc and soleus muscles. So now he was able to put his feet flat on the ground. Yeah.
Thanks, Vivian. That was great. Really interesting stuff. I had some questions about the arm and legs exercise too. That's a commercially available system, right?
Exactly.
And did it have stimulation capabilities, or is that something that you added? And were the arms and legs, was the mechanical coupling forced? Was it hardwired into the mechanics of the system?
Beautiful question.
That's my first question.
Okay, so let me tackle this first question. So when we first started, there were commercially available systems that did arms or legs. And we actually bought a system, paid a significant amount of money, and because the company said, yes, it could do arms and legs. It could do arms or legs. So when we first started, there was no arms and legs systems. There were no arms and leg systems out there that were coupled. So what we did is we cut and paste. We cut the one ergometer from the one and pasted it on an Urges system. So now they were not coupled mechanically. So what we did is we came up with a software system that gave a dial to the participant so that they could now voluntarily and consciously align the arm and leg cycling in terms of the rhythm. Okay? And so we did that in the study. We also had the RTI system that was RT200, which has a mechanical coupling between the arms and legs. And we also used the Berkel bike. So we used actually 3 different systems. And we couldn't tease out the specific differences between them, whether they have an actual mechanical coupling or a conscious coupling. All study participants improved their walking capacity, and it was the sample size was a little bit too small. So now we are not using the cut and paste system. We are using primarily the Berkel bike system because it does not have a motor. So all the work has to be done by the legs with the assistance of FES and the arms working together.
All right. Well, that answers my second question,
Ah.
whether there's a motor assist or not. The 2 examples you showed, pre and post, had different baselines. One was walking in a walker and the other started with crutches. And did you ever see anyone move from a more aggressive assistive device like a walker to become a crutch ambulator? And on a related note, have you thought about a crossover where the first person did legs only and the second person was legs and arms, but they started off as a better ambulator to begin with? So if you put that second person through the arms and legs, and I mean, can you tease out
Yeah.
the effect of the baseline condition, and
Yeah.
did the arms and legs put everybody over
Yeah.
the hump from a walker into crutches?
Yeah. Maybe I can show some slides. Is that okay to just go to some slides? Because what we, just to answer your question a little bit more easily, slides that all took away. There we go. Okay. So this is now the 10-meter walking test for both groups. What you're seeing here is training weeks, change in walking speed, percent relative to their initial walking speed. But the initial walking speed was not significantly different between the two groups. So we were very careful to make sure that the two groups had similar walking speed at the beginning, right? So that we can tease out the difference afterwards. And you can see that both groups have improved in their walking speed, but the arm and leg cycling group improved better. Now, with regards to their assistive device, we actually said you can't change it between testing sessions, because that's going to change the rules, I guess, or it's going to become another variable.
Yeah, they
But
must
we
have
did-
hated that.
What's that?
They must have hated that.
They did, because they would come in and say, look what I can do, and I'm using this, and like, yeah, but you have to use that for the testing session, so that we do not include, would not introduce another variable that we don't have enough sample size, or we didn't at that time, to be able to tease that out. Yeah, but certainly some did graduate, I guess, from a walker to crutches or crutches to cane. Yeah.
And then my third question, and then I'll yield the floor. Everything you talked about was preparatory, right? The walking afterwards did not include TSCS or any kind of stimulation.
Nothing.
So I'm wondering,
Yeah.
could you speculate on adding peripheral nerve stimulation to this to kind of help correct some of the remaining gait deviations, or is there an advantage to using TSCS while you're walking? Do you know any of that?
Yeah. So during the assessments, you're absolutely correct. There was no stimulation of any sort, not FES to the legs, and not transcutaneous spinal cord stimulation, because again, that was going to change the parameters. And these testing sessions are long, so people are not they just want to finish and get on with the training. So do I think that, for example, transcutaneous spinal cord stimulation would improve walking during the act of walking? Possibly, and possibly similar to what, for example, Jiping He and Dick Herman 20-some years ago, with epidural stimulation with people with incomplete spinal cord injury, they saw that you do get faster. After locomotor training, they implanted people with epidural stimulation, and the study participants said that their effort of walking was reduced and the speed of walking was increased. So perhaps transcutaneous spinal cord stimulation would do the same thing. Perhaps, right? Obviously, an implantable system is a little bit easier than something on the back, but that is external. But that could be the case. With regards to peripheral, yeah, absolutely. I mean, what we're trying to see is how far can you improve walking with this strategy as a therapeutic strategy, and then what are the remaining kind of deficiencies that continue to impair walking, and can you augment those with additional stimulation? Most likely, absolutely. I mean, this is not going to make people walk perfectly, but it's improving them in an impressive way, and then we can use other techniques to augment the remaining deficits. Absolutely.
I was very intrigued by the improvements in proprioception that you saw with transcutaneous spinal cord stimulation, and I was curious if you have any ideas about what mechanism might be behind that.
Yeah. We're trying to understand that ourselves, and so interesting. And again, this is very, very preliminary. We actually did two methods for proprioception. One was position matching, and the other approach was, as the robot starts moving your arm, move along with it, matching the speed and the mirrored trajectory, okay? Very preliminary, but with the movement matching, so kinesthesia, we didn't see any difference. So transcutaneous spinal cord stimulation didn't have an effect on that. It seemed that the effect is more on the sense of position. What is causing that is still unknown, but we're trying to kind of eliminate cutaneous input, try to understand how that would affect spinal circuits. So it's still unknown. There's a lot. Transcutaneous spinal cord stimulation is the Wild West right now. So so we're trying to really understand what it's doing. Chet Moritz had a study a couple of years ago that showed that transcutaneous spinal cord stimulation to the cervical cord with the cervical region with rehabilitation improved function, but they looked at single arm improvements. So there's something there. We just need to understand more. So the answer is, well, I don't know. There's more to do to understand this. Yeah.
I'm not from the rehab field, so my question could be a bit naive.
That's okay.
When you think about human studies and you recruit normal volunteers, do you think about recruiting young children who do not have fully developed motor abilities? And you do like stimulation versus pre-stimulation? Do they become better athletes at catching balls and doing things that they otherwise cannot
Very
do?
interesting.
Your IRB might become more complicated, but
Yeah.
your studies
Especially when you're doing stimulation, right? So yeah, I'm not quite sure. It's a weird sensation, right? You get used to it, but you'd need a child to get used to it as well. Implanted systems, and Ron and others here did implants in children many years ago, right? But those were children with spinal cord injury. But the implants are different, right, than putting something on the skin and stimulating through it. But Janie Yang, who's a professor at the University of Alberta, was looking at the development of the CPG in children. And it's not really necessarily the development of the CPG, but studying infants, as young as 3 months old, who don't have completely myelinated corticospinal tract. So it's more like the spinal cord is separated from the brain, and looked at whether they have a developed central pattern generator, and basically held them on top of a treadmill and had the treadmill belt go at different speeds. And these kids were, the legs were following. So the question is, what if you do that, right? So this is without stimulation, at least so that you don't disturb the child or distress them. But what if you put them on a treadmill like that? Would they become better runners? Who knows? All right. Good question. Yeah, that's a good question.
We should probably cut it off here.